My name is Marie Stuber. I'm a postdoctoral fellow in the Nagel lab at the Neuroscience Institute at NYU Langone Health. And the title of my talk today is Sensing in a Dynamic World, What the Antennae of the Fruit Fly Can Teach Us About Sensation and Perception of Movement. So I'd like to start by saying thank you to the organizers for the opportunity to share this work with you today. And in this talk, I will present work which is in part supported by a Brain Initiative K99 or 00 award. To navigate through the world, moving animals rely on sensory cues to guide motor circuits that drive behavior. And in neuroscience, we often talk about from sensory to motor. But of course, the brain, crucially, has to coordinate between sensory and motor systems continuously. So all of these animals rely on this coordination. And in humans, there's evidence that failures in these circuits may result in abnormal behavior and disease. Of the senses, mechanosensation is perhaps one of the most mysterious. It's also very important. For instance, as humans, we use touch to manipulate and explore objects, and caring for young, and to protect ourselves from injury. Yet how displacement at the periphery is translated into a coherent percept is not fully understood. We also rarely passively sense the world. Sensation is often an active process where we move and sense during the same behavior. So how does our brain distinguish between sensations we create with our own actions from those originating in the environment? And what do animals gain by actively moving their sensors in these behaviors? In my research, I use the fruit fly, Drosophila melanogaster, to tackle these fundamental questions about neural circuit function and behavior. The head of the fly, which is shown in the image on the right, houses many specialized sensory systems. For instance, their conspicuous red eyes mediate a rapid visual system. Their two antennae house numerous sensory systems, including a very sensitive sense of smell, thermo and hygrosensing systems, and the largest mechanosensory organ in the animal is in the antenna. These sensory systems are used to guide relatively complex behaviors, for example, to locate distant food sources, during courtship and aggression, and during rapid flight maneuvers. And fruit flies perform all of these behaviors very well in laboratory settings. So to give you a sense of the scale of this organism and its brain, here's the body of the fruit fly, which is about the size of a sesame seed. And its central brain, which is stained in blue here, is about the size of a vanilla bean speck on, for example, in your ice cream. To study these tiny brains, I use many tools. And in particular, I take advantage of huge libraries of publicly available genetic lines. And there's thousands of these lines which are inexpensive to use and easily shipped between labs. With these genetic lines, I can target and manipulate specific subsets of neurons in the brain, for example, by expressing light-activated ion channels for optogenetics, or by expressing fluorescent indicators like the green fluorescent protein, or GFP, shown in this image in green. And using this, I can target small groups or single cells repeatedly across individuals. Using these fluorescent indicators as a guide, I can then measure the intracellular responses from these genetically identified neurons. I record from single cells using a highly precise technique called whole cell patch clamps. And in these recordings, I seal onto the cell body of the neuron with a glass electrode. I break a hole in the cell body through which I record the cell's tiny electrical current. This trace here shows the raw electrical activity or membrane potential from a single central mechanosensory neuron in the fruit fly, and I presented the fly with a sensory stimulus here. And it responds with changes in the graded membrane potential as well as in changes in the frequency of these small amplitude spikes overlaid on top. These experiments are hard, but basically the gold standard for recording neural activity. By carefully gluing intact flies to 3D printed custom holders, I can perform these electrophysiological recordings in intact and behaving flies. So here I'll show you a video of this technique in action. In the top right, we'll see an image of the fly from below, filmed by camera. And I'll play the video now. The trace, the yellow trace on the bottom, shows the intracellular activity or the neural activity from a single sensory neuron in the fly while it's flying. So this technique is incredibly powerful for characterizing neural circuits during the behaviors that they enable. And ultimately, I aim to leverage these and other tools in the fruit fly to discover fundamental properties of neural circuit function, with the idea that these studies may also shed light on mechanisms in other organisms. Fruit flies have long been known to use odors to locate food sources, and we now also know that they use wind to measure the direction from which these odors come from. Wind is transduced at the periphery in flies by motions of the antenna. So here we show the head of the fruit fly again, and we can zoom in on one of the antennae to look at the fine structures in more detail. The antenna in fruit flies, like other insects, is segmented into the first, second, and third segments in fruit flies. And on the fourth segment, there's a passive structure, feather-like structure called the arista. And this acts like a sail to mediate movements of the third segment of the antenna along the long axis in response to air currents such as wind. So how are displacements of the antenna translated into a perception of wind direction? How this transformation occurs in the brain was not clear. So today in my talk, I'll first tell you about how this transformation occurs in the central brain of the fly, and then I'll describe how these circuits function to encode active movements. Studying wind direction encoding, I designed a stimulus apparatus that enables me to deliver the wind to the fly from 5 directions. The fly is mounted in a holder in the center of this apparatus, indicated by the red star in the left image, and I film movements of the antenna with a camera placed below the fly. I'll show you a video of what this looks like here. We're looking at the fly's head from below, and in this experiment, I presented a 4-second tonic wind stimulus of the fly from -45 degrees relative to the midline. I'll start playing the video now, but you may want to keep your eye on the third segment of the antenna and the arista. When the green dot appears, that indicates when the wind stimulus is on. So to my eye, and perhaps yours, these movements are fairly subtle, but I'll show you later that there are neurons in the brain that readily encode these movements. With the help of high school students in the lab, we quantified wind-induced deflections across many flies to obtain an estimate of how these sensory structures, how the antenna themselves encode wind direction. So here I'm plotting the average tuning of the left antenna in magenta and the right in teal in response to wind presented from the 5 directions. And these deflections are relative to the midline of the fly. And as we would expect, motions of the 2 antennae are symmetric. However, we noticed something interesting when we focused in on the tuning of a single antenna, for example, the right antenna here. And what we noticed was that several wind directions could elicit the same antennal deflection, which means that the wind direction tuning of a single antenna is ambiguous. So if an animal was hoping to pinpoint the source, for example, of an odor, it would have a difficult time determining wind direction using a single antenna. However, if we compare the difference between antennal motions simply by subtracting the left from the right, this ambiguity disappears, showing that the antenna together could generate a linear encoding of wind direction. And this linear encoding could potentially be one signal that the fly uses to pinpoint a source of odor or use in other behaviors. So does this comparison happen somewhere in the brain? I mentioned that wind is transduced by motions of the antenna, and these motions are detected by several hundred stretch receptor neurons located in the second segment of the antenna, which together are called Johnson's organ neurons, or JONs for short. These neurons project their axons to a region of the central brain outlined in gray above, called the Antennal Mechanosensory and Motor Center, or AMMC for short. And work from several groups have shown that distinct subgroups of these neurons respond to wind. However, how wind information is encoded beyond this primary processing center was not well known. So to understand how this information is processed in the brain, I first looked towards 2 classes of projection neurons called AMMC projection neurons, or APNs for short, which are anatomically poised to receive wind direction information because their inputs are in the wind-sensitive region of the AMMC. And I hypothesized that these neurons could encode wind-induced deflections of the antenna. To test this, I measured the intracellular responses from single genetically identified APN neurons, and I targeted these neurons by expressing GSP under control of specific genetic lines. And in these experiments, I performed whole cell patch clamp recordings from animals mounted in the same stimulus apparatus that I used to quantify antennal motions. And again, I presented wind from 5 directions. These are the results from the APN2 neuron here in this slide. The anatomy of the neuron is schematized in the top left image, and then a confocal stack is shown below. In the middle, I've plotted the average membrane potential across several flies in response to a 4-second wind stimulus. And what I found was that these APN2 neurons are inhibited by wind from all directions except wind on the ipsilateral side, or the same side of the animal that I recorded the cell on. If I plot the steady-state tuning of these cells now on the right figure across the 5 wind directions for single flies in gray or cross flies in black, I saw a familiar hook-shaped tuning. So now if we plot the average tuning of the APN neurons on the left in blue and compare this to the tuning of a single antenna on the right, we see that they're very similar, and they share this ambiguity in wind direction encoding. So the anatomy of these neurons is restricted to one side of the brain, and so we might expect that the tuning of this neuron is due to its unilateral antennal inputs. But we couldn't assume this because there are neurons that are contralaterally projecting at this level of the brain. So to test this, I blocked inputs from the ipsilateral antenna, shown on the left plot, and this shows that, and in this case, this abolished all wind responses in the fly. When I blocked inputs from the contralateral antenna, there was no significant impact on the wind tuning in these cells. So this tells us that these APN2 neurons are receiving input primarily from the ipsilateral antenna. Are there neurons downstream in the brain that might integrate information from the 2 antennae to compute a linear representation of wind direction? To identify such a circuit or cells in the brain, I looked for higher order neurons that might integrate information from this region of the wedge where the APN neurons project to. So I performed an electrophysiological screen to identify wind-sensitive neurons that received inputs here. And this was a rather labor-intensive screen, but yielded an important discovery, which was a class of neurons that we call wedge projection neurons, or WPNs for short. And the genetic line that we found that labeled these 2 cells on each side of the brain, which each respond in the same manner, appear to be part of a larger class of mechanosensory neurons that run along the same anatomical pathway. So here on the left figure, I'm showing the raw memory potential traces from this cell in response to ipsilateral wind in purple, during which this cell is excited. These cells are inhibited by wind coming from the contralateral side, shown in red. And in the middle, when I plot the average tuning across many flies, we can see that they exhibit a beautiful gradient of tuning to the intermediate wind angles. And plotting the steady state responses on the right, again, it's for single flies in gray and across flies in black, we see that they have a quite linear readout of wind direction. And so the ambiguity in wind direction encoding that was present at the level of single antenna and of the second order projection neurons has now disappeared. Plot the tuning of these WPN neurons on the left in purple and compare that to the antennal motion comparison, we see that each produces a quite linear readout of wind direction. So I guess that the wedge projection neurons indeed are integrating information from the 2 antennae. To test this, I compared responses to flies with intact antennae that are able to detect wind with flies where I blocked inputs from either the ipsilateral antenna or the contralateral antenna. Both of these manipulations result in a reduction in wind direction tuning, but not in a complete loss of response. Further, I found that blocking inputs from both antennae completely abolished the response. So this tells us that the neuron is integrating direction information from both sides and is receiving inputs through the antenna. If these WPN wedge projection neurons combine direction information from both antennae, I next aim to establish where in the brain this happens. Does this neuron directly integrate the information, and if so, where? I pinpointed the likely location of integration from the 2 antennae using several experiments, including dual whole cell patch clamp recordings, precise 2-photon lesions of the contralaterally projecting axons of these cells, optogenetic activation of several putative presynaptic cell types, as well as genetic silencing of these same cell types. So with these experiments, I have come up with an updated circuit diagram for understanding how wind direction information is computed in the brain of the fly. And this is likely an oversimplified diagram, but gives us an idea of how this likely occurs in the brain. So on one side of the brain, antennal movements are first detected by primary mechanoreceptors in the antenna. This ipsilateral information is integrated by the WPN neuron via APN2 and APN3 neurons. Contralateral information is likely provided by B1 neurons and potentially other as-of-yet unidentified cell types that were revealed in some anatomical studies. And this circuit has its mirror symmetric counterpart. And with these experiments, I've worked out what I believe is a sufficient circuit for understanding how wind direction information is computed in the brain of the fly, and it opens up avenues for exploring how wind information is used in the central brain. Next, I wanted to know if there's a behavioral correlate for this, if there's something important about the use of both antennae in an important behavior for the animal. So one important behavior for fruit flies is finding food, and they do this using odors and wind direction, as I mentioned earlier. So for these experiments, I turned towards an apparatus in the lab. These are miniature wind tunnels, walking wind tunnels, which we affectionately refer to in the lab as Manhattan wind tunnels because they have a quite vertical orientation. And I'll play a video of what these experiments look like. We place single walking flies in these arenas, and we present a constant wind stimulus into which we inject an odor, which is indicated when this green light turns on. We can play this one more time, and you might notice that when the odor is turned on, the flies start walking and running upwind. This odor is an attractive odor associated, so there they go running, associated with food. It's apple cider vinegar. And a previous postdoc in the lab quantified this olfactory search behavior. And what I wanted to know was if there's something important about the use of both antennae in this very important behavior for the animal. So for these experiments, I collaborated with a graduate student in the lab, Andrew Matheson, and we placed these single walking flies in the arena. We measured their behavior using real-time tracking and a camera placed above the flies. And in the figure on the left, I'm showing the trace of a single fly in one of these experiments. The black trace shows the 30 seconds before the odor stimulus was turned on. The red trace shows the 10 seconds during the odor stimulus, and the blue trace shows the 30 seconds after the odor stimulus is turned off. In flies with intact antenna, we quantify this upwind run, this red trace here, as an increase in the upwind velocity, which is shown in the central plot. When we block inputs from both antennae by stabilizing them, the flies no longer perform this upwind velocity run because they can't tell from where the odor came from. Then we noticed if you block inputs from one antenna, either the left or the right, flies then perform an intermediate upwind search. And this experiment is in agreement with previous results from tethered flying flies, and it tells us that flies require both of their antennae for robust upwind orienting behaviors. So far in my talk, in this first part, I showed that single antennae provide an ambiguous information about wind direction, but a simple comparison across the two antennae could provide a linear readout. And that integration of information from the two antennae likely occurs at WPN inputs from a sum of APN and B1 inputs. And then I showed that robust wind-based behavior requires input from both behavior. And overall, I described an ascending sensory circuit that translates antennal displacements into a higher order wind direction signal that the animal can then use for wind-based navigation. So now I will tell you about ongoing and future work that I will leverage the sensory circuit that I previously characterized and described in the first part of my talk, and now focus on mechanisms of active sensing. I think it's important to note early now that fruit flies do actively move their antennae. So I'll show you a video here of the fly filmed from the front. In this video, this fly is attached. She's glued to one of these holders, and she's in basically a wind-proof box. And I'm not presenting any sensory stimuli to her. She's not flying in place. So any movements you see of the antenna here are self-generated. Flies produce antennal movements. They can control movements of their antennae using tiny muscles at the base of their antennae. So what are the purpose of these and other types of active movements we observe in fruit flies? Active sensing encompasses a very broad range of behaviors, including any where the animal is actively moving its sensor or emitting energy to alter the types of sensory information it acquires. There are many famous examples of active sensing, including saccadic eye movements in primates, electroreception in weakly electric fish, and whisking in rodents. There are also many great examples of insects performing active sensation behaviors. For example, during trail following in ants, they will actively sample odors on the substrate by tapping their antenna to the ground. Stick insects and cockroaches are great examples of insects that use their antennae for active mechanosensory behaviors. For example, cockroaches have been found to determine characteristics about objects in space based on their, for instance, texture, size, and shape using antennal touch. So they alter the frequency and angle of their antennal movements against the object to do this. Studies in these insects and the classic examples on the last slide, among others, have led to many important discoveries about active sensing, and yet we still don't fully understand this phenomenon. And this is partly due to the complexity of these circuits and accessibility of these underlying circuits. And so what I want to know is, how does the brain accomplish these important behaviors, particularly at the cellular and circuit level? And so here I'm turning to Drosophila as a small circuit model for active sensing to understand these important features of neural function. So I'll show you a couple specific types of active antennal movements that flies perform. And I've observed many different types of antennal movements. I'm showing you just a couple snapshots here. So in this video, we'll see an example of a unilateral antenna sweep. We're looking at the fly from below here. And you'll see her right antenna sweep again. I also observe examples of coordinated movements of the two antennae, like here. I think this looks kind of like a shrug or maybe even an eyebrow raise. And in the next video, I'll show you an example of several types of active movements which are overlaid with passive movements of the antenna. Again, we're looking at the fly from the front here. Halfway through this video, the fly starts flying. And you can probably tell when she starts flying because her antennae are brought into this more frontal position. So she's flying in place here. She's glued to the holder, but her wings are moving, although we're focused on her antenna here. And you may also notice this vibration of the wings, of the antennae, this blurring of the antennae, which is a passive vibration induced by the movements of the wings. And then we also observe these small flick-like movements of the antenna during flight. You'll notice here a couple of flick-like movements. So we see active movements upon active movements combined with different passive antennal movements as well. This frontal positioning reflex that we observe here is observed in all flying insects. However, we don't fully understand what the sensory consequences and function of this positioning reflex is, and I'll come back to this a little bit later. But how do we quantify these antennal motions? To do this carefully and quantitatively, we need to precisely track the different pieces of the antenna. And for this, hand tracking is not feasible based on the volume of data that we have. So instead, I've turned to machine learning tools to automatically track the position of these different features in these videos. So I'm currently using Deep Lab Cut, which is a relatively user-friendly open source package for pose estimation based on transfer learning and deep neural networks. And so to do this, what I do is I hand label a couple dozen frames from these videos. It takes an afternoon or so, and this training set is sufficient to train a robust network for tracking the different segments of the antenna in these videos. And the results are often close, if not better, than human accuracy. So I'll show you that video, the unilateral antenna sweep video again, but now with different features of the antenna automatically tracked using these machine learning tools. So what I can do now is track the individual segments of the antenna, for example, the third and second segments of the right and left antenna, and I can quantify how these different segments move relative to each other during self-generated or active movements versus externally induced or passive movements of the antenna. And I'm currently characterizing the neural encoding of these self versus externally generated antennal movements. So far, I've observed surprising differences in how central mechanosensory neurons encode these different types of stimuli, namely that some cell types appear to encode all movements of the antennae, including both the active and passive components, whereas others appear to only encode externally induced motions or passive motions and remain unchanged during large active movements. So if some neurons in this Wind circuit respond only to externally generated movements, this implies that they must subtract the effect of this self-generated motion. So this signal, so perhaps a subtractive predictive signal from the motor system. And this type of signal carrying information about self-generated stimuli is sometimes called an efference copy or corollary discharge. And here, I will study the cellular basis for this feedback using this small circuit model in Drosophila. To identify the sources of feedback, I'm starting by mapping the motor circuits controlling antennal motions, which up until now were unknown in Drosophila. So I've started by mapping motor neurons at the periphery, and I'm working my way up towards higher order motor circuits that control these motor neurons, controlling movements of the antenna. So to track down these motor neurons and gain access to them to test out hypotheses about predictive motor feedback, I'm screening for genetic lines that label motor neurons, and I'm performing two parallel screens, which each will use these huge databases of genetic lines, and we select candidate genetic lines using very nicely characterized known motor neuron anatomy in larger insects. And we use those to select candidate genetic lines. And then in this first screen, what we've done is we've expressed fluorescent indicators under control of those specific lines, and then we carefully dissect off the antenna and image them. And these experiments are in collaboration with an undergraduate in the lab, Ashley Medina. And through this process, we found several great motor neuron candidates that innervate the first segment of the antenna where the muscles are. In a complementary screen, I'm using an optogenetic approach, and here again I have many candidate genetic lines. And in these I express in the red shifted light activated cation channel crimson, which is a channel reduction variant, and I activate these different circuits with the assumption that if I've labeled or am activating motor neurons or motor circuits, we'll observe some measurable difference in antennal motions. So again, I'm using machine learning tools to automatically track the antenna in these videos, and indeed this screen has yielded several additional great motor neuron candidates that, when activated, induce stereotype deflections of the antennae. And anatomy of the antenna, subsequent anatomy, helps to confirm the identity of these motor neurons. This screen also has the potential to identify higher order motor centers, which we can distinguish from motor neuron candidates using anatomy and restricted to genetic tools. This screen has also identified a genetic line that labels one of the muscles in the base of the antenna, which is shown here. So with the motor neurons identified, I can now go and directly measure the contribution of these cells in sensory encoding by different classes of sensory neurons in this pathway. So for example, do the motor neurons themselves carry a signal that counteracts self-generated movements? This seems feasible based on the particular anatomy of the circuit and the size of the Drosophila brain. And then we can go from here and characterize higher order motor centers and test their potential feedback onto the sensory pathway as well. So using these new genetic tools to target the underlying motor neurons and muscles controlling movements of the antennae, I can directly now test hypotheses about the sensory consequences of active movements. So you may recall that flight positioning reflex in which the flies move their antenna to a more frontal positioning during flight. And what I can do now is use optogenetics to activate either muscles or motor neurons to artificially induce motions of the antennae, and then test what the consequences are of this flight positioning reflex in the encoding of wind direction or velocity by the antenna themselves. And then I can test the sensory consequences of this positioning reflex on the sensory responses of different wind sensory neurons. So that last experiment is a good example of the flavor of experiments I will use to understand why animals perform active sensing, and precisely how do these movements change the information acquired by sensory systems and for what purpose. Using Drosophila, I can precisely connect neural activity and behavior using these powerful genetic tools to nail down these important questions at the cellular, circuit, and behavioral levels. Today, I described how I elucidated a novel mechanosensory circuit that encodes wind direction by integrating information from the two antennae. And then I described several subsequent research areas that will enable me to dissect the mechanisms and complete circuits underlying active sensing in the system. My goal for this work is to shed light on fundamental computations that enable active sensing. So with that, I would like to thank my postdoc advisor, Katherine Nagel, and current and former members of the lab, in particular, and the individuals that directly contributed to the work I showed you today, Andrew Matheson, Sunita Sarkar, Matthew Damiata, and Ashley Medina, several members of the Neuroscience Institute, my former graduate advisor, Michael Dickinson, and my funding sources, including the Leon Levy Foundation and the BRAIN Initiative, which is currently funding me through a K99R00 award through the NINDS. Thank you.